So when Steve asked me earlier this year if I'd like to speak in this series, I had to think quite a bit about that. I wasn't sure what I would talk about, really, because of, as you just heard, I've been doing central administration things for almost the last 18 years and don't have any new data to show you. But after thinking about it, I thought that particularly because I'm actually right now in the process of transitioning back to the department, at the end of June, I will be back with you. That this would provide an opportunity to introduce myself to the many of you who don't have gray hair and have joined us in the last 18 years. And for those who do have gray hair, remind you what I used to do and have been interested in, a topic that you probably haven't heard much about in about 18 years. So we'll see how that works. So I'm going to try to tell you a little bit about how I got into what I did in the past, some of the things that I think we accomplished, how the field has moved since then, and a little bit about where it might be going in the short future and maybe longer future. So we'll see if we can interest some of you in some of those ideas. So just by way of background, remind you that I'll be talking about immune responses, and most of us, I think, are familiar at some level with the immune responses of other animals, of humans, and recognize that they have a number of functions. This is a gross underestimation of the role of immune responses in animals generally, but they certainly are important in detecting and defending against infections in the body. They are a general surveillance system that recognizes and eliminates diseased, damaged, mutated neoplastic tissues in the body, and are the most important component in the recognition of things like grafted or foreign tissue when it's introduced into the body, leading to rejection kinds of responses. In a kind of a 30,000-foot view, there are usually discussed two major forms of immune response in animals specifically. What's called innate immunity, which is essentially that initial response of an animal to recognize, defend against invasions, infections by microorganisms, and it consists of surveillance procedures, surveillance mechanisms, and then the elicitation of defensive responses to try to wall off or sequester the invading microorganism and eventually to kill it. And the kind of effectors that we would typically have in a vertebrate, a mammal, would be circulating blood cells, things like macrophages that phagocytize small particles, constitutive blood proteins that stick to the outside of invaders and initiate complex attacks, basically by other protein complexes like the complement system to lyse bacteria and other kinds of foreign materials, a variety of antimicrobial peptides, and cytokines that recruit other cells to the area where the infection is taking place, where the defense is occurring. And then we have something that for most of us may actually be what we're more familiar with when we think about the immune response, and that's adaptive or acquired immunity, that capacity that animals have to, if you will, let me say loosely, remember when they've been infected by a foreign agent and be prepared so that the next time that they see it, they can more effectively, more rapidly, more efficiently attack and destroy that agent. And this is the system that involves the clonal expansion of cells that produce specific antibodies, for example, that have receptors that interact with a particular invader that the body needs to remember. So you might think about innate immunity as kind of the day-to-day housekeeping kinds of protection, and adaptive immunity as being primed, being prepared after surviving an initial infection to survive the next time that you're attacked. And this is certainly the adaptive or acquired immunity, the things that we talk about in terms of vaccination and things of that sort. So that's kind of background. And now we'll talk about what we know about how the insect fits into this story. And, okay, so start with talking about yesterday. So let's go back into the past. And you've heard that I joined the faculty of the department in July of '77. I came from a postdoctoral research at John Law's laboratory in the University of Chicago. While I was at the University of Chicago, I participated in a large project that the law group had characterizing Manduca hemolymph proteins that were involved in the transport and the metabolism of juvenile hormone. And so that's the link to developmental biology in my past. And the specific things that I worked on there were two proteins from Manduca hemolymph. One is a 30 kilodalton protein which binds juvenile hormone specifically and transports it and protects it in circulation in the hemolymph during larval development, and then a juvenile hormone specific esterase that cleaves off the methyl ester function of juvenile hormone even while it is bound to the juvenile hormone carrier protein. And this appears about mid-5th instar in the tobacco hornworm and rapidly destroys all of the juvenile hormone that's in circulation, so that the next pulse of ecdysone causes the initiation of metamorphosis to a pupa and eventually to an adult. And so I was used to working with Manduca, number one. I was used to working with Manduca hemolymph, number two, and purifying proteins from it. And so as I thought about what I wanted to do as I began my own research group, I certainly wanted to use those skills. And as I read the literature, I became fascinated with some things that I read about the status of knowledge of insect immunity. Now, in general, it shouldn't be surprising to anybody that insects would have an immune response. Clearly, like all other animals, the body surfaces of insects, all the invaginations of the body surface of insects, including their gut and their tracheal system and every other invagination that you can think of, is coated, bathed in microorganisms. And anytime that there is a wound or a puncture to that integument, the microorganism can potentially gain access to the body. We know from things that you've heard from other programs in the department that some insects harbor populations of microorganisms in parts of their gut, for example, to help them digest their food. You know that there are microorganisms that have evolved invasive mechanisms to get through the body wall in various ways, Bacillus thuringiensis through the gut, a variety of viruses or fungi that penetrate the integument of the insect. And so insects are subject to infection at least as much as every other animal. I don't know very many insects that wash their hands as often as we're encouraged to, and so in many respects they have problems that are at least as severe as ours, but yet they're abundant. They've thrived in that environment, and so clearly one would expect that they have some way to recognize and protect invasion of their body or the body surfaces from microorganisms. And so thought was, let's see what we know at this point in time about the protection that insects have evolved against a simple pathogen, a bacterium. And so in this time period, there had already been work on Galeria melanella, the wax moth larva, by June Stevens Chadwick and several other people. Pseudomonas aeruginosa is a kind of a cosmopolitan animal pathogen. There are strains which are very pathogenic to humans. There are also strains which are pathogenic to insects. And so what June Stevens Chadwick did was to take Pseudomonas aeruginosa, inject it into the insect, find out how many bacterial cells it took to kill wax moth larvae, and then she decided to find out whether insects could be immunized. And so She did what we do in immunization experiments with vertebrates. She killed some cells so that they were no longer able to multiply, injected those into the insect first to see if they could prime a response, allowed the insect to wait a period of time, and then injected a dose of the Pseudomonas which normally would have killed the insect, and they found out that in fact you could induce a protected state in wax moth larvae pretty rapidly, actually, within hours, which is much faster than you can in mammals. In mammals it takes days to weeks, and in fact you could very vigorously protected Galeria against Pseudomonas. Now, I thought it was kind of interesting that they picked Galeria as one of the first model systems to study because Galeria lives in a bee city. And a bee city is a pretty clean place relative to many other places that insects live. And so if I was picking an insect that I thought might have a good, strong immune response, I'm not sure I would have picked one that evolved to live in a clean environment. But they did, and it turned out that they found that, in fact, there was an induced protected state. There were some other studies of Galeria in which they injected them with bacteria, and either non-pathogenic bacteria like E. coli that did not mount an infection, a successful infection because they're not a pathogen, or killed bacteria like Pseudomonas, and showed that they induced dramatically increased levels of an enzyme which is known in mammalian immunity, lysozyme. This is an enzyme that lyses the bacterial cell walls of gram-positive bacteria, the peptidoglycan, and they showed that this appeared very quickly with kind of similar kinetics to the rate at which this protected state appeared. And this work was done by Pounding and Davidson, at about the same period of time, maybe a little bit later, a group of essentially microbiologists led by Hans Bohlman in Sweden was doing some work with Hyalophora cecropia, the giant silk moth, diapausing pupae, and did similar kinds of experiments and found that if you inject killed bacteria into them, that again you could develop a protected state pretty quickly that would protect against a much higher dose of viable bacteria, and also found that you could induce the synthesis of some proteins in hemolymph that had biological activity against bacteria. In their case, they were among the first to recognize the production of some antibacterial proteins that turned out to be small peptides that we know as cecropins nowadays. And so that was known at the time. And there had also been some work done with Galeria melanella looking at the role of their hemocytes in this antibacterial response. And they showed, Radcliffe and Rowley showed that injecting bacteria, non-pathogenic bacteria, into Galeria and then sampling periodically the hemolymph of the insects, you could demonstrate a very rapid clearance from circulation of these viable bacteria. Now, at that point, you don't know whether the bacteria are being killed or if they're simply being moved to a compartment that's no longer in part of the circulation. But very clearly there was the capacity of the hemocytes to phagocytize them, because you could look under the microscope and see that there were now inclusions and also large aggregates of hemocytes which melanized and were distributed throughout the body that are called nodules. And I'll show you more about nodules later. So as we were starting, it was clear that there were very rapid clearance mechanisms for viable bacteria that involved hemocytes that sequestered the bacteria out of circulation by phagocytosis and by trapping them in nodules. There was the capacity to induce the synthesis of proteins that appeared in the hemolymph, some of which were bacteriolytic and some of which were bactericidal, and that this all seemed to coincide kinetically with the appearance of a protected state in the insect. Well, Hyalophora cecropia's pupae are big critters, and you can get a lot of hemolymph out of them, and you can potentially do some biochemistry with them. Galeria melanella aren't. And so this looked like an opportunity to use this system that I was familiar with, Manduca larvae, rather than being a diapausing pupa, this is an insect that's wandering around in this dirty world, rather than somebody who's sitting someplace undergoing metamorphosis. And we knew that we were able to get large amounts of hemolymph from the insect, and we could do biochemistry purifying proteins. So this looked like something that we might be able to study in Manduca. So large larvae, increasing knowledge of their hemolymph, should provide an opportunity to do biochemical and cell biological studies, look like something that we might pursue. But we needed a few extra pieces in order for this to get started. One of the challenges is that we wanted to study responses immediately following the first exposure to bacteria. And so we wanted to be sure that the insects that we were starting with weren't already infected, that they weren't already mounting a defensive response. We wanted to develop a system that would have as low a background of infection as possible. And so developing an azenic culture would be one way to do that in the laboratory to minimize the previous exposure of the insects to bacteria. And so that was a challenge. We also wanted to collect large volumes of hemolymph, which we knew we could do. The classic method is to just clip the abdominal horn of the insect, and the hemolymph drips out pretty quickly. But there's a problem, and that is that when insect hemolymph passes through a wound in the body wall, it comes into contact with wounded tissue, which activates the hemocytes that try to plug that wound, and it elicits defensive responses, so that by the time the stuff gets collected in a tube, all of the hemocytes are activated. They glom onto the walls of the tube. They don't stay into suspension anymore, or they clump up, and the whole thing starts to turn black pretty quickly because of activation of melanization reactions. And that obviously was going to get in the way of what we wanted to do, and so we wanted to have a way that we could collect stable hemolymph in order to do some of the studies both with hemocytes and with the hemolymph proteins that we had thought of. And so first thing we had to do was to develop a population of immunologically naive insects, and we did that by developing a set of culture techniques in which we would surface sterilize eggs and then transfer them in a sterile environment onto sterile medium in sterile cups. Every time that the insects had to be transferred, they were dipped in ethanol so that their body walls stayed clean. Never handled them with your hands, and so on. And so we ended up being able to produce a pretty clean colony, so that when you sampled it periodically and then cultured what was on the surface of the insects, you didn't find anything growing. And so had some confidence that they were at least cleaner than they were in their normal lab culture. And the second thing we needed was a technique to collect large volumes of hemolymph that didn't get activated by passing over a wound on the way out of the body of the insect. It turns out that the development of the azenic culture was the easy part. The procedure for developing large quantities of hemolymph took a little bit more ingenuity. Dave Harhoff was a grad student in the lab at the time, one of my first students. Dave was a master's student. He finished his master's degree a while ago. He's now a named professor at the University of Kentucky. Basically, we picked a procedure, we developed a procedure that was modeled after the way that you collect blood from vertebrates by cardiac puncture, which basically means sticking a syringe directly into the heart of the animal and withdrawing it so that it never comes into contact with wounded tissue. And so the way that we adapted that was to take take needles, typical hypodermic needle stock, which turns out to be siliconized on the inside, have it sterile on the outside, break off the hub of that so that the hemolymph can go through the siliconized needle stock without touching plastic, because it turns out that plastic is something that hemocytes recognize and stick to, and insert that needle stock on the dorsal surface of the insect into the pericardial space and advance it a couple of segments past the injection point, and lo and behold, hemolymph drips freely out of that, much faster than it does out of a cut horn, and it also drips for longer so that you can essentially exsanguinate a Manduca larva if you want to. And then you have the problem of what you collect it in. If you collect it in a glass tube, hemocytes recognize glass and they attach to it and they spread and they clump. By trial and error, we found out that polypropylene culture tubes that were sterile did not activate the hemocyte surfaces. And so found out that you were able to, as long as you collected the hemolymph through that cardiac puncture-like procedure into a polypropylene tube, you didn't have to have any phenylthiourea or other inhibitors of phenyl oxidase in your tube, and you could keep it that way for over a day at room temperature. And if you sampled the hemolymph, there were still freely suspended hemocytes in it. They hadn't all stuck onto the wall. So we had a system that would enable us to both study hemocyte reactions and also have stable hemolymph that wasn't melanizing, that wasn't complicated by stuff that was released, for example, from granulocytes and other hemocyte types when they are attacking a foreign surface. And so that turned out to be a particularly valuable advance for us. So with those tools in hand, we were able to do some experiments, and the first thing we thought we needed to do was to essentially reproduce the kinds of observations that had been already in the literature from Galeria, from Hylophora cecropia, and those other systems. And so Dave Drake, Dave Harhoff, Mike Knost, all students in the lab at the time, took on different parts of this project. They were able to demonstrate that prior exposure of Manduca larvae to killed bacteria elicited the same kind of protected state that had been observed in Galeria, that had been observed in Cecropia, allowing survival following injection of an ordinarily lethal dose of bacteria. We are able to measure an increase in serum lysozyme following injection of killed bacteria, and the kinetics were the same kind of time frame as we had experienced for the development of the protected state. Dave Drake did studies on the clearance of injected live bacteria, and he was able to demonstrate that for a non-pathogenic bacteria like E. coli, that there was a very rapid drop, several logs of bacterial density very, very quickly, and then it leveled off and slowly disappeared. As the lysozyme and the other antibacterial activities appeared, the bacteria were essentially completely cleared. If you used a pathogenic bacterium like the Pseudomonas aeruginosa that I talked about before, there was an immediate drop, a slight lag, and then they started to grow because they weren't susceptible to the defenses that were being mounted. And so you could see the difference between a virulent pathogen and something that was non-pathogenic very clearly. By looking at the hemocytes, Dave Drake and Dave Harhoff were able to show that phagocytosis and nodule formation both occurred by the circulating hemocytes immediately after the injection of bacteria, and were sufficient in the kinetics at which they occurred to account for that disappearance of free bacteria from the hemolymph that occurred so quickly in the first hour or so after the bacteria were injected. So basically, at this point, we had demonstrated in this new Manduca system the characteristics that were in the literature for the other insect systems involving Galeria and Cecropia that were in the literature. And so now we were ready to do something new. And so the team staked out their PhD projects like all of you do as you begin to work in a lab. And Mike Knost, Wei Dai, and Xiaoxin Yang, two other students in the lab, through a series of experiments, were able to show that you didn't have to use intact killed bacteria in order to elicit these responses, that you could take pieces of the cell wall of the outside surface of bacteria, like, for example, pieces of the peptidoglycan, which makes up the outer cell wall of gram-positive bacteria. You can digest into fragments so that you have low molecular weight soluble pieces of that, so you no longer have a particle. You have something which is freely in solution. You could eject that, and that would induce the synthesis of lysozyme and accumulation in the naive larvae. And you could do the same thing with cultured fat body from those larvae. You could take the fat body out, put it in tissue culture, add the peptidoglycan fragments, and elicit release into the culture medium of lysozyme with similar kinds of kinetics. So that was something new. We knew now where the lysozyme came from. It came from fat body, or at least that was one of the places that it came from. We were able then to produce hemolymph that was very rich in lysozyme by inducing a lot of insects, collecting the hemolymph, and doing some protein chemistry. And with the help of Mark Hermanson in biochemistry, we completed, back in those days, you still sequenced proteins. You didn't get a piece of DNA and send it over to a lab and the next day get your nucleotide sequence. You actually made fragments and put them through a sequencer and assembled a sequence with overlapping fragments. That's the way we did the Manduca lysozyme sequence. Lillian Dickinson, who was a postdoc in the lab, and Virginia Russell, who just recently retired from Sirius, did some experiments where they killed bacteria and showed that you could elicit the synthesis of cecropin-like peptides. These were the peptides that are antibacterial. They're small peptide antibiotics that had originally been described in the Cecropia moth in the diapause pupa. Manduca makes them too. They were able to show that occurred, and by the way, that also occurs with cultured fat body. And we were able then to do some protein chemistry again, purify the peptides, and we did the sequences for 4 of the Manduca cecropins. And during the same period of time, Xiaoxin Geng, one of the students that was working on hemocytes, was able to quantify the effect of injecting bacteria during this clearance process of the bacteria on the hemocyte population in the insect, and to show that you actually dramatically deplete the population of circulating hemocytes at the same time that you're depleting the population of circulating microorganisms, because those hemocytes are essentially committing suicide. When they form a nodule, when they attack the bacteria, basically they fall out of solution and never go back into it. So actually, that initial clearance process, where very, very rapidly hemocytes remove the bacteria from circulation, in fact depletes the hemocyte population dramatically so that the insect is weakened. And if it didn't have that parallel synthesis of these antibacterial protective proteins, the insect, rather than being protected, would potentially be, in fact, susceptible to another infection. We were able to show that one of the other proteins, one of the other classes of proteins that was originally identified in Hyalophora cecropia, a bacteriostatic protein called atacin, or Addison, also was produced in Manduca. Wei Dai did that work for his thesis. And once we had the sequences for the cecropins and for lysozyme, Lillian Dickinson was able to clone Manduca genes encoding the cecropins, and Amy Molnix cloned the gene encoding Manduca lysozyme so that we could begin to study the expression of those genes. We wondered whether, in addition to these things appearing in circulation, whether these antibacterial proteins might actually be deployed other places in the insect's body where they could potentially be sites of infection. So one of the things that Virginia Russell looked at were the pericardial complex, the collection of cells around the insect's heart, and lo and behold, that tissue, when it was dissected out in culture, produced lysozyme. And as she continued to look, found something which I don't think has been pursued very much since then by any of the other research groups, but showed that antibacterial proteins are actually synthesized and released into the lumen of the gut of Manduca as the insect is beginning to pupate. So as Lepidopteran begins to pupate, the insect first clears its gut, and then it histolizes its gut, because the gut of the adult is a totally restructured organ that doesn't have the same cells in it that the gut of the larva does. During that time period, if there happened to be any bacteria in the lumen of the gut, when the gut histolized, they would be released into the lumen. And lo and behold, it turns out that Manduca has evolved a prophylactic mechanism to protect itself during metamorphosis, to basically cleanse the interior of the gut before it histolizes it, so that it doesn't release infectious agents into the lumen. And Virginia Russell and Tony Bonner did some of that work. About this time, we also wanted to broaden our scope a little bit, and a couple of folks in the lab wanted to take a look at the status of antibacterial defenses in insects in Manduca that were parasitized by Cotesia congregata. Now, the reason that we were interested in this particular situation is that it was very well established by this time that Cotesia is one of the group of wasps that injects, along with its egg, into its host a virus. It's a DNA virus called a polydnavirus, and that those polydnaviruses infect host cells. And it was speculated that they were important in basically suppressing the immune system of the host so that the wasp's eggs would not be encapsulated. And so we wondered, if the immune response to the wasp egg is suppressed, how about the immune response to bacteria? And lo and behold, it turns out that parasitized larvae of Manduca are dramatically more susceptible to infection by bacteria than are non-parasitized larvae, because the virus infects and destroys hemocyte functions so that the insect is no longer able to rapidly clear bacteria from circulation. And if the dose is large enough so that the bacteria begin to multiply before the antibacterial proteins begin to be produced by fat body, the insect will succumb to the infection. So there, in fact, insects that are parasitized by Cotesia are in fact more susceptible to bacterial infection. And with an understanding of what the virus was doing, that became more clear. And so Connie Hallberg then looked at more detail of the properties of the hemolymph of Manduca and the hemocytes of Manduca following the parasitization by Cotesia. And Connie was my last grad student before I decided to venture across the campus and enter the dark side. So this is kind of hitting the highlights of the things that we were able to accomplish in this particular line of investigation. As Larry and others know, we were involved in a bunch of other stuff too, with his lab and with other things, but this was certainly the main line of investigation in the lab at the time. So let's talk about what's happened while we were doing all of that, and then what's happened in the interim to the systems, what we've learned about the immune system of the insect in the interim. So during the same time period that we were doing these things, there a group in France, led by Jules Hoffmann, just opened up the floodgates of information about the antibacterial responses of Drosophila, using all of the tools of Drosophila genetics, and basically told us everything that we knew about the Lepidopteran system and a whole lot more about control systems and mechanisms that were in place, because they could be studied so much faster in Drosophila. And there was a group in Sweden with Bowman, the folks who started doing the Cecropia work. They continued to do that and continued to isolate and identify antibacterial proteins, purify them, characterize them. And so all of those activities continued during the same time period. So by the early to mid-90s, about the time that I was venturing over to the dark side, had my, I lost my lightsaber, and I was big hood. Okay, so what did we know? We knew that every Lepidopteran larva that had been studied, and certainly Drosophila, exhibited similar innate immune responses to initial infection by bacteria that involved both initial cellular clearance and the subsequent induction of the synthesis of antibacterial proteins of various kinds. It seemed like every time you'd pick up a journal, there'd be a new antibacterial protein that was identified during this time period. These innate immune responses accomplished the rapid recognition and elimination of the injected bacteria. At this point, we didn't know much of anything about how hemocytes recognized that these things were bacteria, and how recognition that this was a bacterium as opposed to a latex bead might or might not specifically induce a particular antibacterial response. But They accomplished the rapid recognition and elimination of bacteria by combination of phagocytosis and nodule formation, followed by the accumulation of antibacterial hemolymph proteins, which protected the insect while the hemocyte population was restored by division of existing cells and production of additional hemocytes from hemopoietic organs inside the body of the insect. And so the very simple concept was, hey, here you got a bunch of kamikaze hemocytes, you know, that sacrifice themselves for the body of their insect host, and you know, boy, once they do their job, everybody's going to be really, really in trouble if you didn't have a backup defense. And so at the same time that the kamikaze hemocytes were working, antibacterial proteins started, and so by the time the hemocytes were gone, you had an antibiotic soup there to protect the insect, and that persisted long enough for the hemocyte population to be restored. That's pretty much the model as we understood it at that point in time. And I'll jump ahead and say that we know a lot more detail about that model, but the picture's pretty much the same today. The induced synthesis of lysozyme and various antibacterial proteins and peptide following injection of killed bacteria or fragments of cell wall appeared sufficient to account for this acquired protected state, primed state. June Stevens Chadwick even said immunized state in some of her writings. So that's pretty much what we knew at the time. There was at that point no evidence of anything like an adaptive immune response, a response that had a significantly long memory and enhanced responsiveness to a specific invader that was comparable to the adaptive immune response that we know from mammals, at least in those species. Perhaps this made sense, and I had an opportunity to write a review article for a journal in which I suggested that, you know, all of the insects that had been examined are short-lived. During their lifespan, they move through ecosystems. Maybe all they have to do is survive an infection and escape. So maybe because we were looking at what you might call ephemeral species, that they would be in a particular environment for a short period of time if they were infected and could survive and escape to a new environment. They didn't need to remember who hit them back in the place where they used to live. They just needed to be able to recognize an infection by somebody else and fight it off again and then move on to the next ecosystem. And so you had a Manduca that was, you know, an egg on a leaf and chewing on the leaf for a while, and then going down in the soil and then emerging and flying away, and so on with the other species that had been looked at. And so maybe all insects needed was an innate immune response. Turns out that probably still makes sense for most of the insects that have been studied. But I'll give you a caveat in a little bit. So during the same period of time, turns out that a good friend of mine, or somebody who became a good friend of mine, Rick Carp, who was at the University of Cincinnati, had initiated studies of immune responses in Periplaneta. Now, Rick was a classically trained immunologist. He wasn't an entomologist. And so his way of designing a classic immunological experiment with Periplaneta was to take up an antigen, not an organism, an antigen, and so he picked some protein toxins, inactivated the toxin so that it was no longer toxic but was still the protein sequence, injected it into the cockroaches, and then at varying periods of time challenged those cockroaches with active toxin, with a dose of active toxin which would normally kill the cockroach if it hadn't been treated before. And lo and behold, what he found was that you could induce a protected state with inactivated toxin in cockroaches that would protect them, but that the kinetics of that induction was very different than it was from the short-lived insects that had been studied. It wasn't hours, it was days to weeks. Well, Rick observed that Periplaneta lives about a year, about the same as a mouse. Periplaneta lives in the same environment for that year, like a mouse does. maybe Paraplaneta has a different problem to solve than a short-lived ephemeral insect in an agricultural ecosystem. Maybe there's a reason for Paraplaneta to remember who attacked it before and be able to fight it off more vigorously. And so Rick began to study this some more. Carp's use of protein antigens rather than bacteria more closely models the typical kinds of immunological studies that were done in mammals, and unfortunately, Rick never got around to doing experiments with infectious agents. He continued to do experiments with protein toxins, with protein antigens. So we don't know in the cockroach system, I think even to this day, at least I haven't found it in the literature, what would happen in terms of the induction of an innate kind of antibacterial response in a cockroach that we see in the other systems that had been studied. But what he did find was that if he injected an activated protein toxin, rested the insects for periods of time, then injected a lethal dose that the insects survived, that there was a protected state induced over a period of days to weeks that lasted for a significant period of time and protected against subsequent doses of the same toxin that was used as an antigen, but not other protein toxins. So if you used, for example, a phospholipase, which if injected into a roach is toxic, and then immunized with a denatured phospholipase, and then challenged with that, you found a protected state. But if you challenged initially with the inactivated phospholipase and then used a different protein toxin, there was no protection. So there seemed to be specificity, another characteristic of acquired immunity. When the hemolymph from the immunized roach was analyzed using the classical immunodiffusion techniques that you use to detect antibodies in mouse blood, for example, you take an agar plate, punch some wells in it, put a sample of the antigen that you're using as a test in one well, put a sample of the either control or immune blood in the other well, let them diffuse towards each other. When they reach an equivalence point, there's a precipitin line that forms. The same thing happened with cockroach blood. Precipitin lines were formed, and just like with mouse immunology, if you adjusted the concentrations, the precipitin line moved closer or further away from the wells based on the concentration of the sample that was diffusing from the well. So it had the characteristics that you would expect for the kind of a reaction that we have between antibodies and antigens. Again, the time frame for the protected response was closer to the kind of time frame that you would typically observe in mammalian immunity. So what does this mean? So maybe it means that an insect with a lifespan comparable to a mouse that lives in the same habitat for much or all of its life maybe has a mouse-like problem and has to have a mouse-like solution. Cockroaches don't make immunoglobulins. They have proteins which are cell surface adhesion molecules that have homology with immunoglobulin structures, but they don't make immunoglobulins. So whatever they make is not an immunoglobulin, but seems to have some of the characteristics of it. Unfortunately, when this work was being done in the early 90s, the proteomic, genomic tools that we have available to us today weren't available. I don't know if there's anybody here who's tried to get any significant amount of hemolymph out of a cockroach, but it's extremely difficult. Cockroach hemolymph clots very, very quickly. Getting a tiny drop is a challenge sometime. Rick wasn't a biochemist, he was an immunologist anyway, and so he was basically stumped and wasn't able to pursue the biochemical characterization of the proteins or other materials that were responsible for this effect. So I'm going to come back later, Mike Scharf, and ask if maybe this is something that could be done now. In the intervening years, many groups, the Hoffmann group and many others studied Drosophila melanogaster. By this time, Mike Knost was out on his own, my former grad student, in the biochemistry department at Kansas State with a increasingly large and productive research group. So a lot of work was continuing to be done on Manduca. There are several species of mosquitoes, a number of grain beetles, including Tribolium and Tenebrio, that have been studied now. And we have information that is consistent from all of these sources that the general features that I described for you of Lepidopteran or Drosophila immunity exist in these species. Now there's differences. Some of the molecules are a little bit different. There might be some different antibiotics. Many of them are conserved. Some of them aren't. So what's the picture today? Because of the work of all of those folks on, and particularly Knos's group on Manduca, we understand very clearly that very rapidly after the entry of bacteria or other microbes into the insect hemocoel, a suite of constitutively expressed pattern recognition proteins, which are always present in the hemolymph of the insect, bind to the surface of infectious agents like bacteria or like fungi or like the surfaces of viruses. And the binding of these pattern recognition molecules to the surface of the bacteria marks them. And it is that binding that tells hemocytes that these are things to be phagocytized, that these are things to be attacked, that these are things to be trapped in nodules. So the binding of these pattern recognition molecules, many of which have been isolated and characterized now, are in fact an intermediary that mediates that hemocyte-mediated process that is so important at the beginning. The binding of these pattern recognition proteins also activates circulating hemocytes and changes their adhesion properties, which facilitates the formation of nodules and causes them to be sticky and causes other hemocytes to discharge the contents of their intracellular granules, which is the source of the matrix material between the cells in a nodule that forms, that binds bacteria and forms large clumps. And that pattern recognition also triggers the release of phenyloxidase from a class of hemocytes, which are called enocytoids. And once outside the cells, a cascade of serine proteases clips portions of the prophenyloxidase to produce an active protein and produces active phenyloxidase, which then oxidizes tyrosine metabolites in hemolymph to make melanin that is deposited on the surface of nodules or at the surface of wounds. The combination of phagocytosis and the entrapment in nodule, as far as we know, still accounts for the initial rapid decline in the numbers of circulating bacteria. And here's a picture from Chapman's textbook of nodule formation to show you what's happening. We now know that don't remember which button you told me to push, so I'll point. The pattern recognition molecules binding to the microbe causes the hemocytes to discharge material, which is that little stippled stuff is in the middle, causes hemocytes to grab onto bacteria or things that are small enough and phagocytize them, forming large clumps. The large clumps end up being encapsulated by plasmatocytes the same way that a parasite egg would be encapsulated by plasmatocytes, and the activated phenyloxidase would melanize the whole glump to form little black blobs inside the body of the insect trapped with bacteria. Some bacteria, non-pathogenic ones, are killed in this process. Other bacteria, many examples of pathogens, are not killed inside the clumps. They multiply inside the nodule and then erupt out to cause that rapid increase in the numbers of bacteria that we saw with Pseudomonas aeruginosa infections. We know today that these pattern recognitions trigger also induces the synthesis of antimicrobial proteins, largely from fat body, but also from hemocytes and also in other tissues like epidermal cells. And the serine protease cascade that activates the prophenyloxidase and that's triggered by the microbial components also stimulates the activation of a cytokine named Spatzel, which is a small peptide that was first identified in Drosophila but has been shown to exist in Manduca. And that binds to the cells of the fat body that initiates a pathway for signaling called the toll pathway that leads to the expression of antimicrobial peptides. And there are at least one or two other of those kinds of pathways that also occur in addition to the toll pathway, for example, in Drosophila. The proteinases in hemolymph that function in these cascades are normally kept inactive because of the presence of inhibitors of those proteases that are present in the hemolymph normally constitutively that keep them from doing damage when they're not needed. And the bottom line is that in a recent review by Knost and Nardi, they identified from the Manduca system that we'd studied, where we maybe had a handle on 4, 5, 6 proteins in hemolymph, there now are more than 46 Manduca hemolymph and hemocyte proteins that have been shown to participate in this antibacterial response, and they fall into those categories that are identified below: pattern recognition proteins, antimicrobial proteins and peptides, proteinases, inhibitors, other enzymes, hemocyte modulating proteins, and hemocyte membrane proteins that are involved in binding of signals. And so the Manduca system that we had a small piece in getting started has blossomed largely as the result of the most important product that we produced, Mike Knost. The most important thing that we produce at universities is not papers, it's students. So today, parallel studies in Drosophila and other insect models has revealed similar complex, sometimes more complex control systems, so that the overall picture hasn't changed, but the number of pixels in the picture is really, really increased. And there's a whole bunch more granularity to the structure diagrams. Mike had a slide the other day, and he said, maybe you're going to show this. And I said, not a chance, because it would have filled up the screen with arrows and boxes and little things that are in fact the representation of that granularity that's now available as a result of the work that's happened since I stopped. So what does tomorrow hold? So we've now uncovered, I think, basically all of the molecular building blocks of the innate insect immune response, or have we? Well, it turns out that the answer is probably not, because every time a new species has been studied in recent years, something new has been detected. New molecules, new control mechanisms continue to be uncovered. So we don't know all of it yet, and the availability of tools, proteomics, genomics, bioinformatics, that we didn't have available now makes available systems that we couldn't study before because you couldn't get enough material to do biochemistry with them. So those are all opportunities now, and maybe we can do some things with cockroaches that Rick couldn't pursue. By the way, Rick is not retired yet, so he's available as a consultant. Studies of current models with new probes continues to uncover new phenomena, and just one example of this, there was a 2007 paper by Pham et al. In PLOS Pathogens that documented the existence of what appears to be a new specific priming response in Drosophila that's elicited by injection of a specific bacterium, Streptococcus pneumoniae. And the mechanism that is, the priming mechanism that is elicited by this particular bacterium requires hemocytes, and it requires an active signaling pathway, but it is not blocked by things that block the signaling pathway that we understand so clearly, suggesting that there's another signaling mechanism that can be a priming mechanism. So we clearly haven't answered yet the question of why you get that rapid decline and why you induce a protected state in all species. There's still more mechanism to learn there. And Purdue entomology still has contributions to make in our understanding of these immune responses. You're going to be famous. Mike, working with Ian, is working on a problem in Manduca, looking at the impact of host plant chemistry on immune status. So now, instead of having these nice little clean bugs in their sterile environments, we have somebody asking, well, what really happens in nature? That'd be an interesting question to ask. Isn't nature more relevant than our little sterile lab environment? It was useful to do the sterile lab environment, but let's move on. Perhaps there's an opportunity to re-examine some of Carp's earlier observations. the results that, Virginia Russell and I got just before my lab closed about the existence of antimicrobial responses in Manduca midgut has, as far as I can tell, not been followed up. And so the control mechanisms for this would be anticipated to be developmental controls, not infection-based controls. And maybe there's something else interesting there. And who knows what else? So, we provided the initial characterization of the Manduca sexta antibacterial response here at Purdue. Mike Knost and his colleagues have built that into a vast body of knowledge about the Manduca innate immune response. New research from Ian's lab promises to help us understand what of this might be relevant outside of the laboratory. And the best is probably yet to come. So that's the picture for tomorrow. I want to be sure to acknowledge all of the previous members of my lab whose data gave me something to talk about. And extra thanks to Mike Knost, who over the last couple of months has been shipping me articles to read as I prepared for this seminar. And Mike is the source of the two color pictures that were on the slides. Thank you.